Hi, my name is Nick Zomer. I'm a research technician in the group of Alexander Hoysje at the Department of Human Genetics at the Radboud University in Nijmegen. And today I will be talking about one of the projects we've been working on lately, which is a form of targeted Iso-Seq, in which we combine the expertise of both Samplix and PacBio for the indirect sequence capture and sequencing of large transcripts. At our hospital, we are avid users of PacBio systems, as can be seen when taking a look at our genomics facility. Although, this is already a bit of a dated picture, as 2 of these sequels have already been replaced with 4 new Revio systems, which we now use for many of our long-read applications. One of these applications comes in the form of Iso-Seq, which is now also available in the Kinnex kit. Iso-Seq is basically a long-read RNA sequencing technique, and as this technique uses full-length cDNA transcripts rather than the conventional short-read approaches, we get a more accurate representation of the expressed isoforms, which help us fully characterize the transcriptome. Iso-Seq is something we've been known to use at our department already, as can be seen in this recent paper by René Salz and Emil Forsterveld, in which they studied the transcriptome of pathogen-stimulated white blood cells. Or, for example, this paper by Tabea Riepe, in which she tried to uncover the full transcriptome of the human retina. However, as Tabea also encountered, there are still some limitations to the Iso-Seq method as it currently stands. While the technique will perform great for most use cases, it provides only moderate coverage for lowly abundant transcripts and isoforms, meaning we might not always see back our gene of interest. More importantly, and problematically even, is the bias for transcripts smaller than 10 kb in size. Some genes expressed in the human retina, for instance, are quite large, which resulted in insufficient and incomplete coverage of these transcripts. While the recent paper of Stamerdink et al. Provided an optimized protocol for the sequencing of large transcripts, raising the bar to transcripts of up to 15 kb in size, this is unfortunately still not long enough to get coverage of our largest isoforms. With a relatively new addition to the genomic technologies present at our department, we hope to bring a solution to this problem, the Samplix Xdrop sort. Now, what does this machine do? It makes use of the so-called indirect sequence capture for the targeted enrichment of a region of interest. For this purpose, a small detection sequence is chosen to capture and interrogate the surrounding genomic context. The detection sequence can be placed either in or around a region of interest, depending on the application. And there are many applications for this technique. One might place the detection sequence inside the region of an insertion to make sure it was incorporated into the correct genomic location or whether it found its way to an off-target site. One might place the detection sequence within the unique part of a gene to distinguish between the real gene and the pseudogene. But we can also use this technique to close the gap between 2 known regions or to determine the number of tandem repeats. But then we started thinking, with the concept of Iso-Seq in our mind, what if we would make the detection sequence to be cDNA specific? Would we then be able to capture all the expressed isoforms of a gene of interest? And thus the idea of targeted Iso-Seq was born. In order to test our concept, together with our blindness group, we decided to look into 2 genes with sizable transcripts that are expressed in the human retina, but couldn't sufficiently be covered by conventional Iso-Seq methods. The genes we will be looking at are ADGRV1 and USH2A, both of which are associated with Usher syndrome, an inherited form of deaf blindness. ADGRV1 is a gene located on chromosome 5, which contains 90 exons and a proposed full-length transcript of 19.5 kb. The gene encodes for G protein-coupled receptor, and mutations are associated with Usher syndrome type 2C. USH2A, on the other hand, is located on chromosome 1, has 72 exons and a proposed full-length transcript of 18.9 kb. The gene encodes for a protein important in the development of the inner ear and retina, and mutations are associated with Usher syndrome type 2A. Here, the currently accepted transcript for both genes are shown, consisting of a full-length transcript for both, together with some smaller ones. However, as we have never sequenced these full-length transcripts before, we cannot be sure these smaller transcripts actually exist or are just broken down bits and pieces. Given the size of these transcripts, They served as the perfect test for the targeted Iso-Seq approach, for which I will first be showing off the adapted workflow as to get a better understanding of how this technique works. We begin, of course, with our sample, which consists of cDNA derived from the human retina. In orange, we have the transcripts of our gene of interest. One important note I should make is about the input concentration. While there is a calculator available for the amount of genomic DNA we would need for the other approaches, for our cDNA approach, this is more of an estimate based on how much the gene is expressed in our cell type of interest. Now, to the sample, we add the primers specific to our detection sequence, which is a sequence of up and around 150 base pairs located specifically on the transcripts of our gene of interest. We load this mix of cDNA and primers into our cartridge, which will be placed in the Xdrop sort, where the device is able to compartmentalize the DNA in double emulsion droplets. These droplets consist of a watery phase with a single cDNA molecule and primers surrounded by a layer of oil. In turn, this droplet is again surrounded by a watery phase and another layer of oil to create our robust double emulsion droplets. Now, once we place these droplets in a PCR machine, amplification will take place only in droplets containing the detection sequence and thus a transcript of our gene of interest. Using a special dye, we can color these amplicons in such a way that only the droplets in which amplification took place will show a fluorescent signal. We can load these droplets onto a new cartridge, which we can again place into the Xdrop sort, where the machine will be able to do the sorting process all on its own. For those interested, sorting takes place through a double confirmation of droplet intensity in two separate detection zones. The first detection zone is to establish the presence of a positive droplet. The droplet will pass in front of the laser and then loop back around for confirmation. If the droplet is spotted twice, the flow in the sorting channel will slow down in order to ensure only a singular droplet is sorted. The sorting itself happens in the second detection zone. When the droplet passes in front of the laser here, a valve will open, leading to the sorting well. This channel passes in front of the laser one more time to confirm successful sorting. We can follow the sorting process live on the display of the Xdrop. White dots indicating detection in zone 1, green dots in zone 2, always in pairs of 2 when successful. After the sorting process, we're just left with our molecules containing our detection sequence and thus transcripts of our gene of interest. As we only extracted a few molecules from our sample, we will not have enough DNA for sequencing yet. For this purpose, we will remove the DNA from our double emulsion droplets and repackage them in single emulsion droplets together with some reagents. We repackage the transcripts to compartmentalize the PCR reactions as to avoid bias for easy to PCR regions. This will make sure that amplification works even for the difficult regions. The way we amplify our DNA is through the use of a multiple displacement amplification. For those unfamiliar, a multiple displacement amplification employs aspecific primers that can bind to multiple locations in the genome. Amplification will take place till the newly synthesized strand will bump into an already formed strand, displacing it in the process, through which new DNA becomes available as a template. In the end, this will result in DNA with a branched structure. Due to the aspecificity of the primers, this is considered a whole genome amplification and will also work on our cDNA molecules. After amplification, we will validate whether our capture was successful through qPCR, using primers for the so-called evaluation sequence, which is a small sequence close to our detection sequence. The reason we use a different sequence for validation is to avoid any bias caused by aspecific targets amplified by the detection sequence. In the end, we can calculate our enrichment by comparing CT values of our enriched sample to those of the unenriched sample. Now, of course, we also want to know the genetic makeup of our enriched sample. But before we can send it in for sequencing, a digestion step is performed to get rid of the branch structures resulting from our MDA. After a cleanup of our sample using a custom buffer, we can prepare the sample for sequencing using the standard SMRT library preparation protocol. In this case, samples were sequenced on the PacBio Seq3E system, but of course, this would also work on the new Revio and Vega systems. For those interested, a more detailed version of the entire protocol is available in this paper by Merel Stemerding, on which I contributed to the targeted Iso-Seq portion. Now that we know what the workflow looks like, I can show you how we used it to study the transcriptome for ADGRV1 and USH2A in the human retina. For this purpose, we designed 3 sets of detection sequence primers for both of these genes. One at the 5' end, one in the middle, and one at the 3' end of the full-length transcript. As the molecules in our sample are all different transcripts and isoforms, we expect to capture only transcripts of our gene of interest, and specifically those expressing our detection sequence. For instance, a capture with the front detection sequence of ADGRV1 will lead to the capture of both the full-length transcript and the small front transcript, while the smaller end isoform will not be captured. For this exact reason, we designed multiple primer sets as to ensure capture of the full-length transcript, as well as more accurately define the transcriptome. We performed the entire workflow for both of these genes on cDNA derived from RNA from human retina samples. As mentioned before, we had to make an estimate for the input concentration of the cDNA. But luckily we got it right at the first go, as you can see here for ADGRV1 by looking at the amount of droplets sorted. As the successful range is over 100 droplets sorted per sample, we see great sorting for the front and end capture, while middle lags behind a little bit. This can likely be explained by a lower primer efficiency, as this is the case for both replicates. However, this is not considered an issue, as the other captures should give us all the expressed isoforms. The same can be said for USH2A, where we see great sorting for the front and middle capture, but this time end lags behind a little bit. After the amplification step, validation of capture and enrichment was performed using qPCR. Here we see an ADGRV1 sample on which we performed isoform capture using the end primers. We can see a beautiful difference of roughly 12 CT values between our enriched and unenriched sample, meaning the capture was successful. Again, this is also true for USH2A. Here we are looking at a sample on which we performed an isoform capture using the middle primers. And again, we see a clear difference of around 10 CT values. An additional benefit from using multiple detection sequence primers is that we can already confirm before sequencing whether we have captured any full-length transcripts. As each sorted droplet contains a single molecule, in this example captured with the front primer set for ADGRV1, we know all our captured molecules contain the front part of this gene. If we now were to validate using the middle and end validation sequences as well, and we get a signal, this can only mean they were present on the same molecule as the front part, as otherwise they wouldn't have been captured in the first place. And that is exactly what we can see here. The same can be seen for this USH2A sample on which an isoform capture was performed using the middle primer set. As we also get a signal from our front and end validation sequence, this must mean these regions were present on the same molecule, and thus we have likely captured full-length transcripts. Seeing as how we most likely have full-length transcripts for both genes, we send them in for sequencing. First, we will be looking at this ADGRV1 sample, on which isoform capture was performed using the front primers. The first thing we can appreciate is that we have ample coverage over nearly the entire length of the gene, only leaving some gaps in the middle and front. For all of the exons, we see coverage ranging from 100 to 2500x. Upon closer inspection, we can see the coverage of the gene can be divided into 3 blocks. One from exon 3 to 20, one from exon 21 to 73, and one from exon 79 to 90. Using a PCR on the cDNA, we were able to confirm that the gap between exon 73 and 79 is most likely a mapping issue. The same might also be true for the first 2 exons. Something that is a bit more difficult to explain, however, is the lack of reads spanning both exon 20 and 21. Now, due to the nature of the MDA, we are dealing with chimeric reads, which are reads on which the same sequence is present multiple times as a result of the strands looping back on each other. However, it seems very unlikely that by chance all reads were cut between these 2 exons during the digestion step. Moreover, as these molecules were captured with the front primer pair, there has to be something connecting these regions, as they have to be present on the same transcript. Hopefully, future runs will give us a clearer picture for this gene. The real interesting data comes from this H2A sample, on which isoform capture was performed using the middle primer set. Here, we can see full coverage of all 72 exons over the entire length of the 18.9 kb full-length transcript for the very first time. More notably, we have ample coverage of every exon with a maximum coverage of 200x. While we may still not have a single read that spans all 72 exons due to the nature of the amplification, this is the first time we find compelling evidence for the expression of primarily the full-length transcript in the human retina. If we zoom in a bit, we can see that despite the MDA, we still have reads with decent stretches of subsequent exons. In the interest of time, I cannot show all data, but the technique has demonstrated the ability of detecting a multitude of isoforms and splicing events, and the best way to show this is by demonstrating the capability of possibly identifying novel exons. As can be seen here, a little upstream of exon 22, we can see coverage of the exact same intronic region for multiple reads. This region is present in roughly 15% of our transcripts and has not been previously described. Notably, this coverage can only be seen at the start of each read, and when looking in the UCSC genome browser, we can see this is a highly conserved region with a UTR-like motif. While additional research is definitely still required, this might be an indication that we're looking at a novel exon and potentially even a novel transcript, which is very exciting. So to summarize, we have been able to successfully apply the indirect sequence capture principle on cDNA in the context of a targeted Iso-Seq, which has led to ample coverage over the full length of H2A, mapping reads to all 72 exons for the very first time. This has brought compelling evidence for the expression of primarily full-length transcripts in the human retina, and most notably, the technique has demonstrated to be capable of detecting a multitude of isoforms and splicing events, including the possibility to discover new exons and transcripts. To conclude, this successful test of the technique on ADGRV1 and H2A means that the targeted Iso-Seq can likely be applied on many other transcripts over 15 kb in size, which includes 323 transcripts from 175 genes, making up roughly 0.1% of all transcripts. As such, indirect sequence capture on cDNA adds to both Samplix and PacBio protocols and applications, providing valuable insight into the transcriptome of lowly expressed or large genes. While many more tests and optimizations are to come, the current study already demonstrates the technique's strengths, providing an optimistic outlook on the future of long-read RNA sequencing. And with that, I would like to thank everyone in our department that contributed to this study, as well as the people from Samplix that provided the support, and of course our collaborators at PacBio. Thank you all for listening.